Well, I want to welcome everyone today for this session of our Integrating Bioinformatics Education series. We are fortunate to have Dr. Phil Williams. He is the Bioinformatics Technical Director at the MidSouth Bioinformatics Center located at the University of Arkansas at Little Rock. This center is a core facility. It is designed to help students, faculty, researchers in the state with their computational processing needs and analytical needs when it comes to working with their biodata. And today, Dr. Williams will be showing us how to use Galaxy, which is an open source web-based platform for data-intensive biomedical research. So with that, I will turn it over to Dr. Williams to share his presentation with us today.
Thank you. So with any choice of tools, there are trade-offs to be made. So for Galaxy, then on the upside, there's no programming experience needed. You don't have to be experienced with Linux or the command line or any bash scripting. Also, Galaxy provides the server, so you don't need to have your own server for this. Now on the downside, and what I mean by downside, when I very first did a lecture on this, the Galaxy server was actually down. Fortunately, it came back up shortly during the lecture, so we didn't have any real issues with that. What I've done is make sure that it's running properly today. And so also some of the trade-offs is that for Galaxy, you may have to wait for processing to complete. If there are other people using the system, you may have to wait in a queue until resources become available, and then from the queue it starts to process, and then you wait on the processing. Also, there's limited data storage. However, for the registered users, there is actually 250 gig of storage available. For most bioinformatics processing, that should be very sufficient. So a user could read scientific papers, form theories, find data relevant to those theories, and then apply the Galaxy bioinformatics tools to that data to extract some knowledge from that. They could then use that potentially to write some grant funding and then receive funding to continue with the research. So the landing page is a bit busy here. So what I want to do is cover up the middle of this. And so the middle here that I've covered up is called the workspace. When I switch over to the demo, you'll see what I mean by workspace. So to the left of this workspace, we have the tools. So here is an array of open source bioinformatics tools that can be applied to the data. The data is found on the right side and what they call the history. So they call this on the right side the history. And so typically what we do is to rename that history. So in this example, it goes from unnamed to demo 1. So to demonstrate this intro to navigation and these tools, I'm going to use RNA-seq data as an example. So first we're going to get data sets. We're going to get these data sets from a shared library internally, which is very handy. And in another demo, I will show you how to get data externally into the Galaxy platform. So we'll get some data. We'll perform FastQC on the data. This is a standard tool that is typically used on RNA-seq data to determine the quality of the base calls of this data. So after FastQC, then we perform quality trimming. And so then what we do with the quality trimming data is we align that trimmed data to the genome. So this is not an exhaustive example. This is just enough to get us started so that you can understand the navigation, how the tools work, how to get the tools and the data together. So a little background for RNA-seq data. RNA-seq is essentially the RNA that represents the gene expression inside the cells. And what it's good for is the differential gene expression. between, say, cancer and normal or cancer and a treatment. So what we've got here is the demo with the RNA-seq data, and the star here indicates that I'm going to shift over to the Galaxy platform and start doing the demo by getting some of the shared data. The rest of the slides here are essentially a review of what I'm going to show you. So I'm going to get out of this and move over to the Galaxy platform. Okay, can everybody see this all right? Okay, so what I'm going to do is start getting some data into my history so I can apply the tools to that. So I go to Shared Data under Libraries, and I'm going to use the search. So I'm going to search for tutorial. You can see I've already got that handy. I'm going to go into the tutorial link, and within that, I'm going to go down to this particular data set. So I'm going to click on this link and go in to look at the individual files in this data set. So what I want is to pick up some FASTQ files to process to demonstrate this. This particular data set is what is called paired end. So what we've got is these two files that represent the paired end files. So we're going to get this set of pair, and we're going to get this pair here. So we essentially really have these 4 files that represent 2 different measurements here. So once I click on that, I've chosen these, and I can export those to my history. See how this works. So I'm going to export as data sets. It's going to bring up a window. I've already named my history, so it's going to offer that I can put it into this particular selected history. So all I have to do is hit the import, and we see that we get the import there. And now we can go back to the workspace. So anytime you get stuck somewhere, what you can do is go back to, on the upper left side here, go back to the Galaxy icon, click on that, and it takes you back to your landing page. So we're back on the landing page, and now you can see that we've got some data in the history that we've imported from the shared data. Okay. So the first thing we want to do with this data is apply the QC. We want to determine the quality of the base calling here. So what I'll do here is I'll first bring up the tool. Okay, so I use the search for FastQC. It brings up the FastQC tool. And now you can see that within the workspace, I have the FastQC tool and my data on the right side. So now I need to choose the data. So what I'm going to do is change it from single files to a collection of files. I'm going to select all 4 of these, and then I'm just going to use all the default parameters here. It's already set up with default parameters. I'm going to use those because it's simple for this demo to just go ahead and go with the default parameters. If you have specific needs for your quality control testing, you can apply those here. So once I set the files and the parameters, I can execute. Okay. And so what the green means here is that it successfully put that into the queue. Notice that for some of these, you may not see that that's a clock icon. That means that they're waiting in the queue. Notice that these others are actually running in the queue, and so now they're pending the completion. So you can see that all of these are running within the queue. We're processing this data. So hopefully it won't take too long, and we can actually look at the results. There, we've got some results. So what I can do here is go into this right here. Let's see, I should probably go to, so the eye icon here represents viewing the data. So what I'm going to do is view the data here, view the FastQC web page. And if you have any experience with FastQC, you'll recognize this. So here we have some graphics that represent the base calling. So notice that the error bars get fairly long here towards the end on this side and even on this side. And this is very common for RNA-seq data. where the quality of the base calls kind of drops off on the ends, but is relatively good in the middle. So typically the first thing you're going to do with your data is determine the quality of the base calling. So again, anytime I want to return, I can just go back to my icon on the upper left, and it brings me back to the workspace with the data. Okay. So now that we've looked at the quality of the base calling, we want to do some quality trimming based on the quality values. Typically in RNA-seq data, you've got some data that indicates for each base call their confidence that it is in fact that base. And so what we're going to do is trim some of those that have low confidence on the ends. And this is a standard procedure for RNA-seq data, is to do quality trimming. So the tool, there's several tools for that. The tool I'm going to use here is Trimomatic. If I spell it right, Trimomatic. Apparently I didn't. Let me get my notes here. There it is. Spell it right and it comes up in the search. Very good. Trimomatic. Okay. So I'm going to set this. This is paired in data. So one of the first parameters I'm going to set here is put this as paired in two separate files. Okay. So now we have these two open up for the first of the pair, the R1, and the second pair, the R2. This is classic naming in terms of RNA-seq FASTQ data files. With the ones and the two. So I'm going to go to multiple files, and so this is the R1, so we want to get the R1s in here, and for the R2, get my multiple data sets lined up, I get the 2s. All right, so we have the R1s for the R1 and the R2 for the R2 to perform the trimming. I'm going to go with the default parameters here. For your own data, you can choose to change these kind of parameters. For this demo, it's easy to just come down here and execute that. So we'll see, and again, we've got the green, and the green indicates that it was successfully submitted to the queue, and you see that it's come out of the queue into the run. So it's in the run phase now, and it probably won't take too long. And so while this is running and we're getting our data, feel free to ask any questions. Well, we have our data, but you can still ask questions if you like. So what we're going to do is we're going to then look at some of this data. So let's look at our paired data. So the eye icon here allows us to look inside the data. So let's have a look. All right, so notice here that it says this data set is large and only the first megabyte is shown. Often these kinds of files are several gig, sometimes anywhere from 8 to 10, 16 gig in some cases. They're often very large. It depends on the experiment and the type of sequencing that you get. And so what we can do is we can actually see inside this file. And this is a standard FASTQ format. Our original data was in FASTQ format. And what we've done is trimmed that original data, removed some of the poor quality base calls, and that has essentially done what we call a data cleaning. So this would be depending on how much data was removed, a smaller file. And so ideally what you want to do is get rid of the parts that aren't helping you and have a smaller data set, because it will run faster for you. Better quality data, and it runs faster. Data cleaning. All right, so now that we have the trimming completed, we can move on to the alignment. So let me get my notes here and be ready for the alignment to the genome. All right, I'm going to use, let me get my keywords here. HISAT, yes. We're going to use this tool, the HISAT tool, for aligning the data to the genome. So essentially what's going on here is we've got some RNA-seq data from treatments or controls or comparisons between cell lines, and we want to align that to the genome and find out how much RNA aligns to what particular gene, so we can look at differential gene expression. So let me set up the tools here. We're going to use built-in genome. This is human data, so we're going to find the appropriate human genome to align that to. Let me come down here carefully and get the particular human genome. I think I passed it up. Yes, here it is. I'm going to use the human genome here. And so I have another parameter that I want to set. This is essentially paired in data, okay? And so we want to set our pairs. And so we want this is the R1, so we want the paired R1 here for this one, and the paired R1 here for this one. So we have these two. And we want multiple files here, and we want our paired R2. So I can get my paired R2 and paired R2. Oops, let me get this one, see if I got the, yes, okay. Okay, and we're going to then set this parameter to forward and reverse. And we want to set our paired options here, specify paired in parameters. Okay, and so what we're doing is we're going to go with the default forward and reverse. This represents the method that was used to sequence the data, and so this is paired in. So we want to use these, join these paired in files appropriately as we align this to the genome. And so we're going to go with the default parameters everywhere else, and we'll execute that. Okay, so it's green that it was submitted correctly, and so now we're processing. Notice that we started with paired files, but they're coming together as single BAM files. The BAM files represent the RNA-seq data and they're where they align on the genome. So it's going to represent essentially where they are in terms of the genome and the genes that they align to. And this will then allow us to count how many align to a specific gene. So hopefully we've done this correctly. It may take a little bit of time to process. Typical for an alignment. It's fairly CPU intensive. So while this runs, feel free to ask any questions.
Dr. Williams, we had a question. A participant wanted to know, how do you know which assembly to use? And he's referring to the reference genome.
Yeah, so we happen to know that this was some human RNA-seq data, and so we wanted to use the human genome to align this to. It just happened to know where the data came from. I didn't really go into that much in detail when I picked that particular data set, but that's what we know about this particular data set. And so I think Galaxy is going to have quite a collection of genomes for you to use. But if there's the case where they don't have your particular genome, I believe you could contact them and they may install that genome for you. Any other questions? Very good. So we've got one that's already completed here, and so let's go ahead and look into this. Okay, so it's quite involved. This is a typical BAM file. So it's got this very large header associated with it. Before you get down into the actual, we get down here far enough, we may see, yeah, so I guess, oh, I see we're getting a little bit more data here. Good. So now we've got, notice that we've got chromosome 1 here, and so now we're starting to get the alignment to specific human chromosomes, starting with 1. Probably there's a lot of data here. We may not go past the 1 into the other chromosomes, so we don't want to push that. It may just cause more delay here. So let me just get out of that and move back over to the workspace. And so what I've shown here is that we can get shared data, and I'll show you here before long how to import your own data. We can get data into the system here. We can do the data cleaning in terms of the FastQC to look at the quality of the reads. We can then do the quality trimming to do essentially a data cleaning and reduction step, and then align that clean data to the appropriate genome. Any questions before we move on? Let me come back here and restart this. And so here's where we left off in terms of the slides. And so if I continue in the slides, then let me start here with the data library. I showed you that we went to the data library to get the shared internal data. I used the keyword tutorials, and I clicked on the tutorials link to go inside tutorials. And I went down to the bottom to get this RNA-seq particular data set, drill down into that to get to specific FASTQ files, and notice that I checkmarked the ones that I wanted and then went on to export those to the history. And so I exported as data sets that you saw, and I then had the option to choose the particular history I wanted to put it into, and I went with my named history demo 1, and I hit the import button here, and then it showed how much was the percentage of complete for the transfer, and we have data in our history that we can then apply our tools to. And so I use the FastQC keyword to find the FastQC tool. And here is where I chose all 4 files for that. And so what this does is it documents the specific parameters that I used, in this case mostly the default parameters. So continue on, we see the default parameters in the second half of the page. And what I did was to accept the default parameters and hit execute to process the data. And you saw that we got the green check mark here to indicate that it successfully went into the queue. You can see that it started running, and we got our results, and we were able to look at the essentially the HTML web page results. This is a standard thing. It's been around for several years. Some of you may be familiar with this type of FastQC output, and here we have the error bars showing demonstrating the quality of the reads. And so after the FastQC, you saw me use the Trim-O-Matic keyword to find the Trim-O-Matic tool, and then apply Trim-O-Matic to these. And the key here is to understand that these are paired end, and so you want the R1 first of the pair for the 1 and the 1, and for the R2 the 2 and the 2, and go on to then the other parameters, and you can then click on execute. And you saw that we were able to get that to run. Here we are demonstrating that it's processing the data. So we get our file names that it's going to land in as it's processing the data. And we looked inside, and so this can be important to view the data. We don't want to see this as a black box. It's important to understand what's inside these files and what these files represent. And this is a classic FASTQ data format. And so we then moved on to align reads to the genome. I used the HiSat2 tool. There are other alignment tools. I chose this one. It seemed to work quite well for this particular demo. The trick here is to get the parameter set. Notice that we've got use built-in genome, and so I chose a particular genome. I knew that this was human data, so I chose this particular human genome to align to. And I knew it was paired in data, so I set the paired in data parameter here, and I chose the appropriate files, the R1 paired, R1, and for the R, so this is the 1, the 1, the 1, the 2, the 2 and the 2, for the R2 paired and R2 paired, and on then to the other parameters. Another important parameter is to then set this to match how you brought your data into the system. And what's interesting to me about this is this --FR. What this essentially means for a person who works on the command line is this is the actual switches you would use for the HiSat2 command line tool. So essentially how I view this is that this web page is taking your the parameters from the page, your data, the names of your data and the locations of your data and the parameters you're setting and building a script. It's building essentially a bash script and a submit script for the high performance computer cluster that this is running on and submitting this. It's building the command line tools and then submitting those for you. And so then this kind of gets us down to the execute part of this. I wanted to show that we didn't set anything else in terms of parameters, and we could execute that, and we got that to run. And we got some data out of that. Okay, so what I've done here is to essentially get you started with this. And so after alignment to a genome, you would then be interested in getting the counts of reads aligned to specific genes, and you could then move on to doing differential gene expression analysis on your data. So that's really the end product there, is to determine differential gene expression. Okay, any questions on this before we move on to another demo? All right, if there's no questions, then I'll move on to the second half, the proteomics side of this. So this is a different Galaxy server. This is the Proteo RE proteomics server. It has a different list of tools here, proteomic related tools. And the data that we're going to look at is essentially some mass spec data in terms of of how they determine the proteins of interest here. And this is the link to the tutorial I used to develop this demo. In this demo, I noticed that they would process the data essentially one at a time. And what I wanted to do was see if I could process this data 3 files simultaneously. So we'll see how all that works out. So a little background for this, where this data set comes from. This is proteomic characterization of human exhaled breath condensate. And what they're interested in is identifying proteins secreted in the respiratory tract and from the lungs. And so it seems fairly relevant here recently, where we're somewhat concerned about viruses and what people are shedding in terms of through the lungs and through saliva and other means. So don't worry, we're not going to do all of this. This is just kind of an overview of some of the processing methods. What we're going to do first is import the data from the source. In the previous demo, we used internal data. For this demo, we're going to use external data. I'll show you how to import your own external data. So your own in terms of if you have your own FASTQ files, or if you have an external source of FASTQ files. I'll show you how to introduce those into Galaxy so you can process that data. So just like we did with the RNA-seq data, we're going to need to do some data cleaning. So in this example, we're going to filter out what's called technical contaminants. So for this, the definition of the technical contaminants from this mass spec data is that it fell below a certain a fixed threshold. They had certain values that they determined. If it's above these values, they have confidence in the data. If it's below that, it's noise, and they don't really have confidence that it's relevant data to their study. So I'm going to demonstrate some filtering techniques on this. And then after that, another filtering technique is to look for biological contaminants and then filter those out as well. So first, the technical contaminants based on the threshold, and I've got a list of these that I'll show you here very soon, and then on to the biological contaminants. So they're only interested in the proteins from the lung, and so you can imagine when they get this kind of these samples through this device that people are breathing into. There's also the opportunity to get proteins associated with the saliva. And so they don't want these saliva proteins in this analysis. So the trick is to find a way to extract those. And so first then I'm going to show you how to get the external data into Galaxy. So let me come over here to my page. I think I'm through with this one, so I'll just go ahead and log out of this one to get it out of the way. And let me come over here to this one. And so what we want to do is come into Get Data, and we're going to upload from the computer. All right. And specific looks like I left that one open. Okay. Let me reset that and do it again. Let me reset that. All right. So we want to do you can see I was practicing, and so I'm going to then do what's called a fetch data. So what I need is the URLs of where this data resides out in the world. So let me show you the page that I use to get that. So let me come over here to my notes, and I'm going to get this URL, and I'm going to come over and I'm going to then open up this real quickly and show you the original source of this data. So this is the original source of the data for this particular study that we're looking at. And so let me show you how you would get data from the web relevant to what you want to analyze. So I'm going to, for this, I'm going to right click on this link. I'm going to poke the A key, and I'm going to come back over here to my notes and come down here and demonstrate that what I've got is essentially the URL for downloading this data. Notice that at the end of this, I don't know if you can see it here, if the font's big enough, there's the question mark download equals 1. We can use that, but it's not very pretty for the data. So I'm going to pick this up and put this into, paste it right here as one of these things that I'm going to pick up. I'm going to hit enter. I'm going to show you where I get the rest of it. I was a little concerned that if the page of the source was down, I wouldn't have a good way to demonstrate the collecting the data here. So I also have copies of this on a web page in the cloud. So I'm going to show you how I get those particular URLs related to that. So these are the two others that I'm interested in. And so I just paste those right in. Let me just bring this up so you can see all three of them. Yes, so you can see that we've got the original source to pull down the data, and we have an alternate source for the data as well. Okay, in case the main source was offline. So now that I've put in the URLs that you would use to download this data, I can then hit start. And so 100% there means it's launched properly. That's good, and you can already see that it's running in the queue and it's downloaded that data. So now what I've done is demonstrate to get data from external sources and import it into Galaxy. Okay, any questions up to this point? So now what we want to do is to filter out some of the, well, let's look at the data first. I think that'll be relevant. So we'll look at the data here. See what it is. So these are UniProt IDs. So these are protein IDs. From the mass spec data, then they determine these mass values related to specific proteins, and it generated a list of protein IDs, these UniProt protein IDs. So this is our source of data that we're interested in based on these UniProt IDs. So what we want to do now is some data cleaning. We want to filter out the protein IDs that are below this threshold that we're interested in. So what I'll do is I'll then use a particular tool, filter by keyword. So I'll come over to the tool here and filter by keyword, filter by keyword. So in my search then I brought up this tool that I want, filter by keyword or numerical values. So I can filter by keywords or by numerical values. You notice that in the data we didn't have any numerical values. We only have essentially words, protein IDs. So that's what we'll use. So I'll come over and go from single file to multiple files. So I'll collect all three of these. Okay. And so the operation we want to use here then is we want to, let me get my notes lined up. We want to discard specific keywords. So what I'm going to do is set that to from keep to discard. Okay, and so now what we're going to do is going to go filter by a keyword. Okay, so we're going to launch this one, and we're going to then let it know where these keywords are located. There's only one column, and so we're going to go with column 1. That's the only column we have. And we want to then determine, we want to put in the keywords that we're going to use to decide what to extract, what to discard from this data set. And so I've got a list of these protein IDs that I'm going to put in here in my notes, which are right here. And you can see that my list is these protein IDs right here. So I can paste that in. And so what we've done now is we've set it to discard. We're going to look into column 1 and discard anything that matches this keyword. And we're down here, and we can now execute. Okay, so now we've launched that. It's in the queue waiting for resources. It found resources. It came out of the queue into the run state. It's now running, and these files are fairly small, and so it runs very quickly. So now what we can do is look at our data again. So again, we've got this one column of data, and so essentially what we've got, if any of these keywords we wanted to extract were in this file, they're not in this filtered file. they've been removed. And if we look at the discarded for this one, what's come out of this one, we can see that these particular protein IDs were found in that file, and so were removed from the filtered set. So I'm going to go ahead, since we've already looked at the discarded lines and we're not really interested in the discarded lines, I'll go ahead and delete that and delete this one to sort of simplify things. Okay, so what we've got is we've got the filtered. And one more discarded. Yes, one more discarded. Okay, good. Okay, so now we've got the new filtered set. Okay, so the next thing we want to do is we want to do an ID conversion. What we're interested in now is filtering out the contaminants. So that means we have to figure out what is a contaminant here, and that means to know what tissue this protein comes from. We're interested in lung-related proteins, but not salivary gland related proteins. So that means we need to determine what tissue these are coming from so we can find the ones that are salivary gland and remove those. So I'm going to then go to convert proteins ID. Let me see if I get my tools lined up. Okay, ID converter is the keyword I'm looking for here. ID converter. And so for the ID converter, looks like it only takes human, mouse, and rat at this time. There may be others later on. So when I choose this tool, I can start setting my parameters. Okay, and so I can choose the files. Notice that we've got the file here. I'm interested in processing all three of these at the same time. So I'll choose all three of these filtered files. And then we've got our IDs still in column 1. So we go with column 1. And let me just check my notes here and be sure I'm on the right track. And we've got, so the source that we're using to convert these to a gene ID is the protein ID. So we keep that as it is. And now we're going to set the output that we want. So we have the default output here. And what we're interested in is getting the ensemble gene IDs. So what we're going to do here is we're going to layer on more data. We're going to do, previously we did a data reduction, and now we're going to do a data enrichment step. We're going to start adding things to this data, specifically the gene IDs, so we can use the gene IDs to then find the tissue that it comes from, so we can remove things that are not lung related. So I can go here, we've set the parameters, and I can execute that. Green, so it's in the queue waiting to run. It comes out of the queue, it's in state. And so while it runs, let me know if you have any questions. We've got some data already, okay. So let's go ahead and look at this set, they're all in. So now let's have a look. So this is the data enrichment step. We started with protein IDs. We then filtered those to a smaller protein ID set. We've used protein ID to find gene IDs, and now we want to use the gene IDs to find the tissue expression. So the tool I'm going to use is add expression data. So I'll come over here and use my keywords, add expression data right here. So I want to, before I go any further, I want to point out that anytime you feel like you get lost here, I don't know if I've shown this previously, I think I skipped this particular step. Anytime you feel like you've been off on the wrong track or lost somewhere, you can always come back to the upper left side and click on the icon here, and it returns you back to your starting page here. And so now we can go ahead, now that we've cleared things up, and say, well, you know, I kind of got confused. Let me just start over here. And so I can then go again and, oh, yes, I want to add expression data. And so I can pick up my tool again, say, okay, I'm on the right track. And so what we want to do is we want to use the ensemble gene IDs as a source for adding the expression data. And so the question is, what column was that in? Do we remember what column the expression data was in? So that was in column 2. So I'm going to go here and set that to column 2. Previously, our data set only had one column, so it made it simpler. But now we've done data enrichment. We've added a second column, and that second column contains the ensemble gene IDs that we're interested in. We're going to use that to get the gene expression. And now we're going to set the particular data sets. Okay, and so we're going from single to the multiple. And so I can go ahead and do all 3 at the same time. It seems to run just as fast with 3 as it does with 1. And let's set our parameters here in terms of our output. And so we want to get some specific data out of this. We want to continue with the gene name. We want the gene description. We also want the RNA tissue category, and we want the RNA tissue specificity. And those are the 3 that we're looking for. Okay, so this is going to help us to do more data cleaning in terms of removing the proteins associated with salivary gland. So, and keeping the ones that are lung oriented. Okay, so go ahead and execute that. So it's green, means that it went into the queue. The clock here indicates, all right, so it's out of the queue into the run state. That's always good. And so probably won't take too long to process this. Yes, very fast. And so now let's look and see what we've added to this. Okay, so we'll view data. Okay, so now we've added quite a bit here. So we have our original column we started with. We've done some data enrichment to add the gene ensemble IDs. Now we've added several more columns based on what we checkmarked earlier in the tool. And so now we can move over here and see some other columns that we've added to this. And so the important thing here is that we've got tissue here. And so what we can do is a simple search. Oops, I'm in the wrong place. Let me see if I can get into here and do my search. I'm in the wrong spot. I can try and show you a search here. Let me see if I can get that to work. Oh, here we go. It's working now. Yes, I didn't do enough keys. So what I'm searching for is the keyword salivary. Okay, it may show it better if I finish my search. That was what I did wrong. I didn't give it enough words. Okay, so here we've got lines. We've got rows in this data set that contain the keyword salivary gland. This is exactly what we need. We need to have this to the file in order to do the next filtering for essentially the data cleaning step. All right, so what I've demonstrated here is that we can import outside data, and we can start doing data cleaning and data enrichment on our data sets. And so I'm not going to, I can't really finish this one completely because it would take a little bit longer to process this. So let's go back to the slides and do a quick review. So what I did was demonstrate that we can get external data into our history. And I showed you that we use this paste fetch function specifically. And I've included here the URLs that can be pasted into this for downloading the data. And you can see that it's pasted in here. Once you get them pasted in, you can do the start and get your data in. So it's all downloaded, and it's essentially started downloading these, and you can see that it's still running the download process here, and then it completes, and we've got our data. And so I demonstrated that we wanted to do this this filtering. We wanted to remove these technical contaminants from our data. What we used was the filter by keyword or numerical data tool for that, and we used a search tool by name to find that. We then set the parameters appropriately and executed that. So we pulled all three files in at the same time and then set these other parameters. We set it from keep to discard. We wanted to discard those. And then we set our filter keywords. And you can see that where I previously plugged in our keywords for filtering and discarding. And so you saw that that successfully went into the queuing system, and we've got it started, and we've got completed data for that. And then we have files containing our filter data, and I showed you that we had discarded data as well. Okay, and so then we go on to look at salivary proteins, and what we did was convert the protein ID to gene IDs, and so that we could then filter out the proteins that were to salivary gland. Okay, and so here is the parameters that we set. We chose the filtered data, and we set our columns, and we chose the ensemble gene IDs for that. We got that data processed, and then moved on. And I showed you that we had data enrichment, where we've added columns to the data. And then we needed to know what column to use here. And so we know that it was column 2. Our gene IDs that we're using for this are in column 2. So we have our data, and we want to use column 2. We want to specifically check the type of data that we want to come out of this process, and because we're interested in the tissue specificity of this for the step of continued data cleaning. Okay, and so we've got some data from that. Okay, and so what I showed you then is that the data comes out has in fact some of this salivary gland keywords that we want to use to move forward for the data cleaning step. Okay, and so what I've done is to get you this far, and you can then do the self to complete this. Here is the link for the tutorial I used to build this onto a kind of a slide-based demo set for teaching. And so then we come to the end of the slides. Any questions on any parts of this?